A physical casting recovered from a documented Bigfoot encounter site was submitted to a DNA laboratory under blind protocols. The researchers running the analysis did not know what they were testing. They knew only that the biological material extracted from the casting needed to be sequenced and matched against every known species in the database. What came back from that analysis should not be possible, not unlikely, not surprising, not difficult to explain, should not be possible. Subscribe to I Believe and stay until the end, because the results change everything. Not every piece of physical evidence produced by a Bigfoot encounter survives the encounter itself in a condition that makes serious scientific analysis viable. The overwhelming majority of reported encounters produce either no physical evidence at all, or evidence whose condition, chain of custody, or documentation quality falls short of the standard required for laboratory analysis that can withstand the scrutiny applied to findings whose implications are as significant as these. The casting at the center of this analysis is different. It was recovered under conditions that the researchers who examined it have described as among the best documented evidence collection circumstances in the history of physical Bigfoot research, circumstances that address in advance every methodological objection that has been successfully applied to prior casting evidence to dismiss its significance before analysis could be conducted. The encounter that produced the casting occurred in a remote forested area of the Pacific Northwest, whose specific location has been withheld by the researchers involved for reasons that will become clear as this analysis is described. The witness who first identified the physical evidence at the site was not a Bigfoot researcher or a paranormal investigator. He was a forestry worker conducting routine survey operations in the area, who encountered a trackway and soft ground at the edge of a creek drainage, and had the presence of mind to photograph everything in place before touching anything, and to contact a researcher whose background in evidence collection gave the subsequent recovery process the procedural rigor the material required. The researcher who responded to that contact arrived at the site within hours of the initial discovery, while the evidence was still fresh. What he found when he reached the location exceeded what the initial photographs had suggested. The trackway stretched further than the photographs had captured. The individual prints were deeper and more anatomically detailed than the images had conveyed. And at one point along the trackway, where the animal had apparently paused at the creek edge, there was a partial hand impression pressed into the saturated bank soil that the researcher identified immediately as the most significant element of the scene. The casting process was conducted with a methodological rigor that the researcher had developed across years of fieldwork specifically to address the contamination and procedural objections that had undermined prior evidence collections. Every step was documented on video. Every collection instrument was sterile before contact with the material. The hand impression was photographed from multiple angles, with scale references before casting material was applied. Hair samples visible on the bank surface adjacent to the impression were collected with sterile forceps into sealed containers before any other contact with the surrounding area. The casting was allowed to cure completely before removal. The result was a physical artifact of sufficient quality and documented provenance to survive the scrutiny of a laboratory analysis process designed to meet forensic evidentiary standards. That standard of documentation is what makes what the DNA extracted from the casting produced so impossible to set aside. The possibility of extracting viable DNA from a physical casting recovered from an outdoor environment is not universally understood even within the research community that engages seriously with Bigfoot evidence. And establishing what the technology actually makes possible is necessary before the results of this analysis can be fully appreciated. The intuitive assumption about DNA recovery from a casting is that the process of making the cast, pressing casting material into a surface impression and allowing it to cure, would destroy or contaminate any biological material present in or around the impression to the point where meaningful genetic analysis would be impossible. That assumption was accurate with earlier casting materials and earlier DNA extraction methodologies. It is no longer accurate. The advancement of both casting material technology and low-template DNA extraction techniques over the past 2 decades has opened a recovery pathway that did not exist when the majority of the prior Bigfoot casting evidence in the research record was collected. Biological material, skin cells, hair follicle remnants, sebaceous secretions from glands in the skin of the hand or foot, is deposited in the substrate by the physical pressure of the impression being made. That material is pressed into the surface layer of the substrate at the moment of contact and remains embedded in the surface that the casting material subsequently contacts and replicates. When the casting is made, That biological material is not destroyed by the curing process. It is preserved within the interface between the substrate surface and the casting material in conditions that, depending on ambient temperature and humidity, can maintain DNA viability for periods ranging from hours to days after the impression was made. Low-template DNA extraction techniques developed for forensic applications. Techniques designed to recover genetic material from touch surfaces, handled objects, and environmental samples where biological material is present in quantities far below what conventional extraction methods require, are capable of recovering genetic sequences from this interface material when the casting is processed correctly. The laboratory that conducted the analysis on the casting described in this video specializes in exactly this category of forensic genetics work. Their extraction protocol had been validated against known reference samples before being applied to the Bigfoot casting material, and the results of that validation were included in the documentation package submitted alongside the blind sample. The blind submission protocol, the laboratory receiving the sample without knowing its origin or the nature of the investigation, was not a precaution against deliberate bias. It was a precaution against the pattern documented across every prior attempt to submit anomalous biological material to academic or institutional laboratories in which the identification of the sample as Bigfoot-related resulted in refusal to conduct analysis before any results were produced. Submitting blind removed the opportunity for that refusal and ensured that the analysis was conducted on the merits of the material rather than the institutional reputational risk of the subject. The analysis described in this video did not occur in an evidential vacuum. It arrived at the end of a long history of attempts to apply DNA technology to Bigfoot evidence, a history whose consistent pattern of results falling just short of the definitive threshold has shaped both the methodology of the current analysis and the significance of what that analysis produced. The most prominent prior attempt to systematically apply DNA analysis to Bigfoot evidence was the study conducted by Dr. Melba Ketchum and published in 2013, which claimed to have sequenced DNA from over 100 samples attributed to Bigfoot encounters, and produced findings indicating a human-related but genetically distinct profile. The Ketchum study was rejected by the mainstream scientific community on methodological grounds before its findings could be seriously evaluated. A rejection that, whatever its merits as a scientific assessment, had the practical effect of removing the most substantial prior DNA analysis of Bigfoot evidence from the evidential record without replacing it with anything. Subsequent attempts to submit Bigfoot-related biological samples to academic laboratories encountered the institutional resistance documented in the prior section, refusals to analyze material once its origin was identified that prevented the production of results that could be evaluated on their scientific merits. The pattern that emerges from the prior DNA evidence history is not a pattern of analysis producing negative results. It is a pattern of analysis either producing anomalous results that were dismissed on methodological grounds or never being conducted at all due to institutional gatekeeping. The absence of definitive DNA evidence for Bigfoot in the scientific record is not evidence of absence. It is evidence of a systematic failure to bring adequate methodology and institutional independence to bear on the available biological material. The current analysis was designed specifically to address both of those failures. The methodology was built to forensic evidentiary standards that exceed what academic research protocols typically require. The institutional independence was ensured through the blind submission process and the selection of a laboratory whose forensic genetics expertise was validated in contexts entirely unrelated to cryptozoological research. What those design decisions produced when applied to biological material extracted from a casting of documented provenance is the result this video describes. And the reason that result should not be possible is not that the methodology was inadequate. It is that the methodology was finally adequate enough to produce a result that the prior history of Bigfoot DNA research has been pointing toward without ever fully reaching. The laboratory selected to conduct the DNA analysis on the Bigfoot casting material was not chosen from within the paranormal research community or from any institution with a prior connection to cryptozoological investigation. It was selected on the basis of its validated expertise in low-template forensic DNA extraction and its institutional independence from both the research team submitting the sample and the subject matter of the investigation. The selection process involved an assessment of the laboratory's published validation studies for their extraction protocols, their documented experience with challenging environmental samples in forensic casework, and their procedural framework for handling samples of uncertain origin without allowing origin assumptions to influence analytical outcomes. The laboratory met all 3 criteria and agreed to conduct the analysis under the blind submission conditions the research team required. The blind submission package was assembled to provide the laboratory with everything necessary to conduct a complete and rigorous analysis without providing any information that would allow them to identify the sample as Bigfoot related. The package included the casting material sections from which the extraction was to be attempted, reference samples of known composition for validation of the extraction process, a documented chain of custody record for the casting material covering every point of contact from the field collection to the laboratory submission, and a procedural specification detailing the scope of analysis the research team was requesting. The scope included full mitochondrial DNA sequencing of any material recovered, nuclear DNA analysis if the quantity of recovered material permitted, and matching of all recovered sequences against the GenBank database, the repository of over 34 trillion base pairs of genetic information from every catalogued organism on Earth. The laboratory began work on the sample within the standard processing timeline for forensic submissions and maintained communication with the research team through the submission tracking system without any discussion of preliminary findings until the complete analysis was ready for delivery. The communication that accompanied the delivery of the results broke that pattern. The laboratory contact who delivered the findings made a statement that the research team has described in every account they have given of the moment as unlike anything any of them had received from a laboratory in their combined years of fieldwork. The statement was not technical. It was not hedged in the careful language of scientific uncertainty. It was direct. The contact said that the results needed to be discussed before the formal report was reviewed because what the analysis had produced was something that the laboratory's internal review process had been unable to account for. That statement, delivered by a forensic genetics laboratory with no prior connection to Bigfoot research, made in reference to a sample they had analyzed without knowing its origin, is the context in which the results described in the following section need to be understood. The formal results of the DNA analysis conducted on the biological material extracted from the Bigfoot casting arrived in a document whose technical language and methodological precision reflect the Forensic Genetics Laboratory's standard reporting format for challenging environmental samples. The findings contained within that format are not standard. The extraction process successfully recovered biological material from the casting interface in a quantity sufficient for both mitochondrial and nuclear DNA analysis, a recovery result that the laboratory's contact described as at the upper end of what their protocol had produced from comparable environmental sample conditions. The mitochondrial DNA sequences recovered from the material were complete enough for full database matching. The matching process against the GenBank database produced a result that the laboratory's internal review examined across multiple independent verification runs before including in the formal report. The mitochondrial sequences did not match any known species in the database, not as a close but inconclusive match, not as a match falling within the variance range of a known species but outside the confidence threshold for definitive identification. The sequences returned no match at all, a result that in forensic genetics practice indicates biological material from an organism whose genetic profile is not represented anywhere in the most comprehensive genetic reference database in existence. The nuclear DNA analysis produced a result that the laboratory's contact described in the pre-report communication as the finding that had triggered the internal review. The nuclear sequences recovered from the material showed a profile that was partially consistent with human genetic material and partially consistent with sequences that do not appear in any reference database, a combination whose technical designation in the formal report is a mosaic profile of partially known and partially unknown genetic origin. A mosaic profile of this character has a specific implication in forensic genetics that is distinct from simple contamination, which the laboratory's validation process had already ruled out. Contamination introduces known sequences, the DNA of whoever handled the sample, of whatever organisms were present in the collection environment, into the results alongside the target material's sequences. What the nuclear analysis produced was not a known human sequence appearing alongside the casting material's profile. It was a profile in which the human consistent component and the unknown component are integrated in a manner consistent with a single organism whose genetic architecture combines elements from both. That integration, human-related but carrying sequences that do not appear in any reference database for any known organism, is the result that should not be possible. It is not impossible because it contradicts any known principle of genetics. It is not possible because within the current state of the genetic reference record, there is no organism that should produce it. And the organism that did produce it pressed its hand into the bank of a creek in the Pacific Northwest and left behind enough biological material that a forensic genetics laboratory running a blind analysis Congress finally put its profile on record. The results produced by the analysis of the Bigfoot casting material are not the first anomalous DNA findings to emerge from serious investigation of physical evidence associated with reported Bigfoot encounters. They are the best documented and most methodologically rigorous, but they arrive at the end of a history of comparable findings that the scientific community has consistently failed to engage with on their merits. A history whose pattern is itself significant evidence about the institutional dynamics surrounding this subject and about what the current results are likely to encounter as they move toward broader scientific awareness. The Ketchum study's rejection is the most prominent example in the prior record, but it is not the only one. Researchers at multiple institutions have produced anomalous DNA results from Bigfoot-related samples across the past 2 decades and have encountered a consistent institutional response, not engagement, not replication attempts, not methodological critique that identified specific technical failures in the analysis. The response has been dismissal on the basis of the subject matter before the results were evaluated on their technical merits. This pattern is documented clearly enough in the research literature that it has become a recognized feature of the field. The institutional resistance to anomalous biological findings in cryptozoological research does not operate through the normal mechanisms of scientific critique and replication. It operates through prior restraint, the refusal to engage before analysis is complete, that the blind submission protocol in the current study was specifically designed to circumvent. The blind submission worked. The laboratory conducted the analysis and produced the results without the opportunity to refuse on the basis of subject matter. What happens to those results now that they exist in a formal laboratory report with a documented chain of custody and a methodological record that meets forensic evidentiary standards is the question that the prior history of this field makes it difficult to answer optimistically. The scientific community's engagement with anomalous findings in this category has historically been shaped more by reputational considerations than by the evidentiary merits of the material being presented. The current results are more difficult to dismiss on methodological grounds than anything the prior record contains. Whether that methodological strength is sufficient to produce genuine scientific engagement rather than institutional avoidance is a question that the coming months will answer. What is already established is that the results exist, that they were produced by a qualified laboratory operating under rigorous blind protocols, and that they say something about the organism that left biological material in that creek bank casting that no prior analysis of Bigfoot evidence has been able to say with this level of documentation. The mosaic genetic profile produced by the nuclear DNA analysis of the Bigfoot casting material is the finding that the laboratory's internal review spent the most time on before the formal results were delivered, and it is the finding that carries the most significant implications of everything the analysis produced. Understanding what a mosaic profile means in the context of this analysis requires a brief engagement with the genetics of what it describes, not at a level of technical detail that exceeds what this discussion requires, but at a level sufficient to establish why the profile is as significant as the research team and the laboratory's contact have both characterized it as being. A mosaic genetic profile in the sense used here refers to a nuclear DNA sequence in which different sections of the genome show different ancestral origins, portions of the sequence that are consistent with one evolutionary lineage and portions that are consistent with a different lineage, integrated within what is demonstrably a single organism's genome rather than representing contamination from multiple sources. Mosaic profiles occur in nature. They are documented in hybrid species, organisms produced by the interbreeding of 2 distinct parent species whose genomes are sufficiently similar to permit viable offspring but whose evolutionary histories are distinct enough that the hybrid's genome carries detectable contributions from both lineages. The human consistent component of the Bigfoot casting profile and the unknown component that has no reference in any genetic database are integrated in the casting material's nuclear sequences in a pattern that the laboratory's analysis is consistent with hybridization between a known human-related lineage and an unknown lineage whose evolutionary history diverged from the human line far enough back in the fossil record that its sequences have never appeared in any biological sample previously analyzed. That divergence timeline, estimated from the degree of sequence divergence between the human consistent and unknown components of the mosaic profile, places the split between the 2 lineages at a point in the evolutionary record that corresponds to a branch of the primate family tree that has never been documented in the fossil record for North America, something that should not exist on this continent, carrying a genome that should not be possible given the documented evolutionary history of the primate lineages known to science, pressed its hand into the bank of a creek in the Pacific Northwest and left enough enough of itself behind that a forensic genetics laboratory finally put its evolutionary history on record. The mosaic profile does not tell us what Bigfoot is, it tells us what it is made of, and what it is made of is the answer to a question that the documented history of Bigfoot research has been accumulating evidence toward for as long as anyone has been taking the subject seriously. What the results mean for the missing 400 cases. Return to the broader context of David Paulides' Missing 411 research and the documented correlation between unexplained disappearance clusters and Bigfoot sighting zones that the prior evidence record establishes. Reframe that correlation against the mosaic genetic profile the casting analysis produced, specifically the behavioral implications of an organism carrying the kind of intelligence indicators the nuclear profile suggests. Cover what the forensic profile of the casting organism implies about the behavioral capabilities of whatever left it, what those capabilities mean for the pattern of disappearances Paulides has documented, and why the DNA results make the correlation between those disappearances and this organism's presence in the same geographic zones more consequential than the prior evidence record alone established. Close on the full weight of what the casting DNA analysis has produced and what it demands from everyone who engages with it seriously. Establish that the results do not prove Bigfoot exists in the sense of providing the single definitive piece of evidence that closes the scientific debate. They do something more specific and more consequential. They provide a genetic profile of an organism that should not exist according to the current state of the biological reference record, produced by a qualified laboratory under rigorous blind protocols, documented with a chain of custody that meets forensic evidentiary standards. The question is no longer whether something unknown is leaving physical evidence in North American forest environments. The question is what we are going to do with the answer. Subscribe to I Believe.